In this video, we will continue our discussion of the characteristics and classification of viruses. Topics addressed in this video include capsid morphology, viral shapes, the viral envelope, and finally viral classification. The capsid is a protective protein coat of a virus. The capsid is made of protein subunits called capsomeres. Capsomeres may be made of a single protein or a combination of multiple types of proteins. The proteins of the capsid interact with one another to protect the viral nucleic acid while outside of the host cell, and then disassemble or uncoat to release the nucleic acid once inside the host cell. The viral nucleic acid contains the information needed to have the host cell synthesize capsid proteins for new virus particles. The capsomeres of a viral capsid can be arranged to form three types of shapes: helical, polyhedral, and complex. The helical viruses have capsomeres that interact with one another to form a tube around the nucleic acid. The image in the top left is the tobacco mosaic virus, which is an example of a helical virus. There are many animal viruses that have helical nucleocapsids within an envelope. Such viruses include the measles, mumps, influenza, and rabies viruses. Polyhedral viral capsids are roughly spherical in appearance. The most common type of polyhedral capsid is the icosahedron, which has 20 sides with 12 evenly spaced corners. Some viruses synthesize the icosahedral capsids from a single type of capsomere, while others use several types of capsomeres. Complex capsids have many different shapes. An example of complex capsids is seen in the bottom two images. The image is of a T4 bacteriophage. The T4 bacteriophage capsid has an icosahedral head to package the viral nucleic acid. It has tail fibers and a base plate that interact with the surface of the bacterial cell, and a sheath through which the nucleic acid moves as it is injected into a host cell. Viruses do not have cell membranes because they do not have the machinery to synthesize them. However, some viruses are covered by a lipid membrane known as an envelope. The envelope is derived from host cell membranes by a process called budding. We will examine budding in more detail in our discussion of replication of animal viruses. The viral envelope may be acquired from a variety of membranes within the host cell, including the cell membrane, nuclear membrane, or membranes of the endoplasmic reticulum. When viruses acquire envelopes, they must have a way for the membrane to interact with host cells they are going to infect next. They do this by inserting viral proteins into the membrane before exiting the host cell. The proteins that are inserted into the lipid bilayer are known as glycoprotein spikes. Viruses lacking an envelope are referred to as naked, while those with a membrane are called enveloped. Envelope viruses must have glycoprotein spikes. Viruses are not included among the classification schemes of living organisms. Virologists, however, classify and name viruses to aid in their study and identification. Viral classification is done by the International Committee on Taxonomy of Viruses, also known as the ICTV, which was established in 1966. To date, they have classified over 40,000 virus isolates into orders, families, genera, and species. Virologists classify viruses based on their nucleic acid type, capsid morphology, presence or absence of a lipid envelope, and the size of the virus. Table 13.2 lists some of the viruses that infect humans. Notice that the primary means of classification is nucleic acid type.